Okay, so it's topic 12, DNA technology. And there are a lot of terms that I have listed here. Lamar, could you confirm that it's recording?
Yes.
Okay, thank you. So these are the terms that you might have heard a lot in some shows, or you might have heard other people talking about. So what are these? So let's start with the broadest term that you have heard, biotechnology. So even if I don't define what's biotechnology, just use your common understanding of bio plus technology to explain what do you understand by biotechnology. I don't need a very scientific definition. I just need a common understanding of what do you understand by the word biotechnology? Anybody?
Technology that has to do with life.
Yes, any technology that uses biology, right? That means any technology that uses living organisms or cells or tissues or organ systems, any kind of biological systems, that is technology. So when we are using technology that uses biology, that is biotechnology. Now biotechnology is the broadest term. Then the second term is DNA technology, which is part of biotechnology. DNA technology is when you are using a technology that uses, instead of any biological system, it is using specifically the DNA, the genes. So whenever there is any kind of genetic modification or any kind of change, editing, addition, deletion, whatever, that uses DNA, then that is DNA technology. That means DNA technology is part of biotechnology. Biotechnology is this big umbrella. DNA is a smaller umbrella under the big umbrella. Now within the DNA technology, you will hear recombinant DNA technology or genetic engineering. That means it's an engineering that plays around with your genes. That means we are causing a change in the DNA, and that's why the name recombinant. Recombinant means you're doing some kind of recombination. You're changing the combination, the permutation and combination of the DNA, and you're using this as a technology. So that is recombinant DNA technology, or in other words, genetic engineering. Then there are so many different types of DNA technology that are in use these days. Okay, the most common one, genetically modified organisms, GMO. All the corn, soybean, anything that you get in US is all genetically modified these days, right? It has to be, to give us high yield, to make them resistant to diseases and insects and pests. So that is all genetically modified. GMO bananas, GMO corn, GMO rice, GMO soybean, most of the agricultural products that you find on the grocery market counter, these are all GMOs. In addition to genetically modified organisms, what other kind of DNA technology or genetic engineering are we witnessing these days? Second one is DNA profiling. You know, in any kind of criminal case in courts these days, DNA evidence is used, right? For example, some kind of blood sample was found in the scene of a murder, and under the nails of the victims there were some skin particles that the victim, as the victim was fighting for their life, they tried to save themselves, and some skin samples were obtained, and then DNA is extracted from those skin samples, and if they have suspect 1, 2, 3, 4, whatever, then they try to match the DNA obtained from the crime scene with that of all the 4 suspects, let's say, and whoever it matches with, then we know that is the murderer or the criminal or whatever, right? So that is DNA profiling, when we are identifying the DNA, and we use it mostly in forensics to prove somebody guilty, prove somebody innocent. So this was DNA profiling. And then human gene therapy. Human gene therapy, a little bit of it is used or has already started being used for medical applications to cure diseases, certain very few genetic diseases. But if provided the correct funding for research, human gene therapy could open so many avenues and doors for diseases that so far medical science has not been able to treat. Only we control the symptoms. And how all of this is done, these are some of the common techniques that are used, very, very basic. Every molecular biology genetics lab uses this. First is you obtain the DNA, this is called DNA extraction, from any sample. We can obtain DNA from blood, urine, nails, skin, hair, saliva, whatever. Any DNA that has, any sample that has cells, you can obtain DNA from it. Can we obtain DNA from red blood cells, Sean? No, red blood cells don't even have nucleus and chromosomes. How can we obtain DNA from red blood cells? Then after DNA is extracted, we use a technique called PCR, polymerase chain reaction. You will read about this in the slide. And this is used to make a lot more DNA than what we get, right? So for example, I want to see, if this DNA matches, and I got very little bit of the skin sample from the crime scene. So then how do I use that DNA? It's just a minute amount, very, very small amount. So then I use this technique to make copies of that DNA. So it's like from that little sample, from where I got maybe like this much of DNA, then I can make this much of DNA using PCR. This is a DNA amplification technique where we increase the amount of DNA. And finally, we want to see the DNA. How do we see? This is molecular biology level. So we need to be able to visualize something. And that's when we use this technique called gel electrophoresis, where we can visualize DNA as bands. So today's lecture, we will be watching a lot of videos in order for you to understand. When I was a student, we didn't have access to all these YouTube videos. So we just had to read what was written in the books and try to imagine it mentally, kind of. It was more difficult. But nowadays, with all these, very, very cool and awesome videos, you can see all this animation, it's much easier. So let's start with the first video.
Recombinant DNA technology is a technique in which a fragment of DNA from a donor cell or organism is isolated and then inserted into the DNA of another cell or organism. This allows scientists to introduce a new characteristic into an organism by inserting a new gene into it. Recombinant DNA technology involves 4 major steps. Firstly, a DNA fragment containing the gene of interest is obtained from a donor cell. Secondly, a suitable plasmid is obtained from a bacterium. Plasmids are commonly used as vectors to transfer the gene of interest into a host cell for expression. Thirdly, the DNA fragment containing the gene of interest is cut using an enzyme called restriction enzyme. This enzyme acts like scissors. It recognizes a specific base sequence and cuts the DNA at a specific point. On the other hand, the same restriction enzyme is used to cut open the plasmid. Lastly, the DNA fragment containing the gene of interest is inserted into the open plasmid with the help of another enzyme called DNA ligase. This enzyme acts like glue. It catalyzes the joining of the DNA fragment and the plasmid. A recombinant plasmid is finally formed. This recombinant plasmid can then be introduced into a host cell for different purposes, such as producing proteins of other species like human insulin, or producing genetically modified organisms that possess new characteristics.
So this was just the first basic video. We will be watching more videos which will explain the same philosophy or principle again. So these are some of the terms, the definitions given here, and I've already explained, what falls under what. So biotechnology, which is the broadest term, is the use of DNA technology for studying and manipulating genetic material. And as you know, that most of all the agricultural products that we are using nowadays, they are all genetically modified organisms. Now, genetically modified means organisms that have one or more genes from outside. That's not their own. Now, that gene, if it is from the same species, it is still a genetically modified. If it's from a different species, then that organism is called a transgenic organism. So, for example, let's say scientists are trying to find a cure for some kind of cancer. They want to do an experiment. They cannot introduce a new gene into human beings and make them act like guinea pigs, right? So what they would do is first try to understand the effect of this gene by causing these modifications, introducing first the human cancer-causing gene in a mouse. so that the mouse starts expressing the human cancer gene. So now we have created a transgenic mouse because that mouse DNA contains human genes, and the mouse and the human are two different species. So the mouse is now a transgenic mouse model that is very commonly used in all of the molecular biology labs. If you see this this particular picture, these are fish which actually glow in the dark. These are glowing fish. How were they created? They were created in lab by scientists. They took a gene which codes for a fluorescent protein. This was obtained from jellyfish. You guys know that jellyfish glow in the dark. So jellyfish have this special gene that can make them glow, that produce a protein, and the protein glows in the dark. So scientists took this gene from jellyfish and added it to the DNA of these fish. So they have created a transgenic fish, and this fish, because now it contains the glow gene, this also glows in the dark, right? So this is all has been done using recombinant DNA technology. And recombinant DNA technologies are used for cloning, gene cloning. Now you heard the term plasmids in the video, which was taken from a bacteria where a gene was introduced. What is a plasmid? A plasmid is a small, round chromosome that bacteria has, which is not its main chromosome, but it's still a chromosome, and it is very easy to manipulate. It's very simple. So it's very easy to use it as a genetic material in labs for experiments. So most of the times, when a vector has to be created or when a recombinant DNA technology has to be used, so let's say I want to introduce a human gene into the mouse, how do I take that human gene and put it in there? So first what I'll take is I need some genetic material where I can input the human gene. So I'm going to take a plasmid from a bacteria, take my gene of interest, we call it the GOI. So let's say whatever the GOI is for me is P75. So I take the P75 gene of interest, I put it in a bacterial plasmid. So the bacterial plasmid is this round DNA. I have put in my human gene there. So now this contains the human gene. And now this is ready, this plasmid, recombinant plasmid, we call it the vector. It is now ready to be transplanted or injected into a host cell. It could be a cell, it could be an animal, whatever. So now I can inject it, and then that particular animal or that particular cell will start expressing the gene of interest that I have inserted into the plasmid. Makes sense? So let's watch another similar video.
Many years ago, diabetics who needed insulin to survive had to inject cow or pig insulin into their bodies. This was not ideal for many reasons. Today, the insulin that diabetics inject is human insulin, and it is produced by microorganisms such as the E. coli bacterium and certain strains of yeast. But how do these microorganisms produce human insulin? Genetic engineering. Scientists are able to turn certain microorganisms into mini factories that make useful substances that improve our health, the environment, and the economy. But what is genetic engineering? An organism's DNA makes up its genes, which code for all the proteins the organism needs to survive. Each gene codes for a different protein, or part of a protein. Genetic engineering is the manipulation or changing of the DNA of an organism. Genetic engineering involves removing a gene from one organism, which is called the donor, and transferring it to another organism, which is referred to as the recipient. The recipient is called a transgenic organism, or a genetically modified organism. There are two basic purposes of genetic engineering. Sometimes, we require large volumes of a particular protein to be made. Scientists use transgenic microorganisms to produce large volumes of this protein. For example, insulin, growth hormone, or vaccines. An organism, B, can have a gene from a different organism, A, introduced to give it, B, some advantage that A has naturally. Organisms like B are called genetically modified or transgenic organisms. For example, a gene that manufactures a toxic chemical in a bacteria can be introduced into a maize plant to make the plant toxic to certain insects that would eat it, such as caterpillars. Let's now look at the process of genetic engineering. How do you transfer a genetic characteristic from one organism to another? although it's a very complex process, it can be summarized rather simply. We'll use the example of how the insulin gene is inserted into a bacterium in order to manufacture large amounts of human insulin. The gene that codes for human insulin is found on chromosome 11 at position 15.5. The insulin gene is cut from chromosome 11 using special enzymes called restriction enzymes. Bacteria possesses small circular strands of DNA floating in their cytoplasm. These strands of DNA are called plasmids. A plasmid is extracted from the bacterium cell. The plasmid is cut open using the same restriction enzymes that were used to cut the insulin gene from the human chromosome. The restriction enzymes leave sticky ends where one of the two DNA strands is slightly longer than the other. Because the same restriction enzyme was used to cut both the human DNA and the bacterial plasmid, the sticky ends are complementary and will allow for joining using complementary base pairing. The insulin gene is joined to the plasmid by the sticky ends. An enzyme called ligase is used to complete the joining of the two pieces of DNA. The modified plasmid is then reinserted into the bacterial cell. The modified bacterium is a genetically modified or transgenic organism because it contains some human DNA along with its own bacterial DNA. Its bacterial DNA is called recombinant DNA. Bacterial DNA recombined with human DNA. The bacterial cell is placed in a fermenter, which allows for rapid asexual reproduction in ideal conditions with the optimal temperature, pH, and lots of food. Because the bacteria reproduce asexually, the offspring are all clones of the original transgenic bacterium. They all have identical recombinant DNA. The bacteria express their own genes in order to survive. In other words, they make their normal bacterial proteins, but they also express the human insulin gene and make human insulin. The fermenter has become a culture of mini factories, all producing human insulin. The insulin is then extracted, purified, and packaged. Humans can now inject themselves with human insulin, and that is how genetic engineering works.
If you liked the video, give it a thumbs up.
Did this make sense? Now, do you guys have an idea of how human insulin is made? Now, anytime you see a patient injecting themselves with the insulin, you know exactly how it was made. A human gene was identified, which makes the insulin protein. That gene was removed, okay? So a cell was extracted, that gene was removed in the lab using restriction enzymes. So now that particular gene has sticky ends. Then the scientists would take plasmids, they use the same restriction enzyme to cut it. So the plasmid also has sticky ends. So this gene, insulin gene, and the plasmid are joined together. Now the plasmid contains the human gene that's going to make human insulin protein, and then that recombinant plasmid is then reintroduced into bacterial host cell. And we take the bacterial host cell, if you take into those pharmaceutical industries, you see these big fermenters, big cylinders in which millions of bacteria are growing and multiplying and reproducing, and as they produce also human insulin protein, and then that insulin protein is extracted, purified, bottled, packaged, sent out to pharmacies, and this is where you get your accumulate. Now this is using recombinant DNA technology to make useful products. For example, you see here, insulin is provided to people with diabetes. What else is made? Protein for dissolving clots. You know, sometimes people have a clot that is circulating in their system, which can be extremely dangerous, can cause them, give them stroke. So then there are medicines or proteins which are used for dissolving clots. There are genes for cleaning up toxic waste, because we human beings keep polluting earth, water everywhere that we go. And so then we need these to clean up whatever toxic spill we've caused. Genes for pest resistance in our agricultural products. So all of these, a genetic engineer identifies the gene of interest or GOI that codes for a valuable protein such as a potential new drug. So this is the process that is being explained, which you already saw in the videos, cutting and pasting DNA. Now we move to a very, very new hot technique, which is called CRISPR-Cas9 system. You see the CRISPR-Cas9. So when I was a student, when I was doing my PhD, at that time CRISPR-Cas9 was not that well known. It was only when I had completed my PhD and I was doing postdoctoral research, that's when CRISPR-Cas9 started becoming commonly used. And the people who invented this, who gave this information to the world, they got a Nobel Prize in physiology or medicine for coming up with CRISPR-Cas9. Because the advantage that this technique has in gene editing is the cell doesn't have to be extracted out of the body. The cell can still be living in the body while you do this technique, and gene editing can be performed in living cells. We don't need to have the cell, taken out and then again re-injected. So basically the mechanism is that this particular Cas9 protein cuts any DNA sequence that is complementary to a molecule of RNA which is combined with the Cas9, and then the RNA is complementary to the DNA sequence. Let's watch this video.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAN. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
So, CRISPR-Cas9 has totally revolutionized, the medical field, because nowadays, imagine theoretically now, if you can see, of course, research is always very scarce, a grant is always scarce, so scientists cannot do as much research. So let's say a particular gene has become defective due to a mutation. And we know that this particular defective gene is giving the patient this kind of disease, some kind of neural disorder or whatever. The patient is paralyzed because of that. But the researchers, scientists, doctors have already identified the defective gene. So how can the patient be cured? If we can get rid of that defective gene, right, and replace it with a healthy gene, how do we do that? It's a living patient. It's not a cell culture or a transgenic animal that I'm treating with. So what we do is we use this system. So Cas9 is a protein. It's an enzyme that can cut, okay, DNA. And Cas9 comes with, so scientists, what they can do is they can design an RNA that is complementary to the defective DNA sequence, right? So this Cas9 RNA, guide RNA complex is injected into the patient cells. So the cells can go and can be done in the stem cells. So it will go, and this guide RNA will look for the matching DNA. Where is the matching DNA? In the defective gene. So it goes and due to complementary base pairing, it will pair with the defective mutant DNA. And as soon as the guide RNA has paired, now the Cas9 protein cuts that DNA. So once the DNA has been cut, the defective gene has been cut. So the defective gene is cut, it's removed from the DNA. And addition to the Cas9 and the guide RNA, a new healthy version, correct version of the gene is also inserted. And the correct version of the gene goes and inserts itself in the broken part where the defective gene was. So we have not only removed the defective gene, we have replaced it with a healthy, normal version of the gene. So now the patient can be cured, and no wonder that the scientists who came up with this received the Nobel Prize. That's the highest reward that a scientist can get. So gene editing is, CRISPR-Cas9 method. Now, medical applications of recombinant DNA technology, you already saw Humulin, which is the human insulin that is being made. You look at these big fermenters. This is where the bacteria are grown and multiplied. This is where human insulin is produced. Of course, very sterile conditions, highly sterilized. This is a transgenic goat, okay, that carries a gene for an enzyme called lysozyme. Lysozyme is an enzyme that is found in our tears, that is found in our saliva, anything. So as soon as you put a food, whatever food you're eating in your mouth, can you guarantee that the food is free from any germs?
No.
However well you might have cooked it, or however well you might have washed that fruit, right? There's always a chance that when you put food in your mouth, there are a lot of thousands of microorganisms that are entering along with the food. So how will the body protect itself? The body protects itself at various stages. From the moment the food has gone into your mouth, the saliva comes, right? You start salivating and the saliva mixes with the food. So the function of saliva is not only to break down food and make it into small pieces so that it's easily digested, but also to protect you. So there is this lysozyme enzyme that will come and that will mix with the food. Lysozyme is antimicrobial. It kills bacteria. Not only that, anytime something goes into your eyes, what happens? Tears come out. What's the main purpose of tears? Well, apart from heartbreak, then there is this washing of the eye. Washing of the eye is a protective mechanism that your body is using. It could be dust, it could be debris, it could be a small, bug that has flown into. As soon as you get something, you close your eyes, natural instinct, you don't have to think about it, right? So that no more entry of foreign things. And then tears come out. It's going to wash the eye. What does tear contain? Is it just water? No.
It's solution.
It's a solution of things. It has lysozyme, antimicrobial enzymes. The eye is trying to protect itself, right? So now in some people, the gene that codes for this enzyme, the gene that codes for this protein becomes faulty, defective. So that means their body cannot make any more lysozyme. That means now they have less immunity, less protection. So scientists have come up with this. This goat is a transgenic, carries a gene for an enzyme called lysozyme. So then what will happen is this goat will make a lot of lysozyme enzyme. That lysozyme enzyme can be extracted from this goat's whatever body fluids, right? And then that can be packaged, bottled, can be used as a medicine to supply to give to those people who are not making lysozyme enzyme. So this is all genetic engineering, recombinant DNA technology. These are things that are being